Today's speaker is our beloved and our very own Dr. Bobbi Pritt. Dr. Bobbi Pritt is a professor of laboratory medicine and pathology at Mayo Clinic in Rochester, Minnesota, and serves as the chair of the Division of Clinical Microbiology, and she's the medical director of the Clinical Parasitology Laboratory. Dr. Pritt completed her medical training at the University of Vermont College of Medicine, followed by a residency in anatomic and clinical pathology. She then completed a fellowship in medical microbiology at the Mayo Clinic. She also holds a master's degree in medical parasitology from the London School of Hygiene and Tropical Medicine in England. Dr. Pritt's main areas of interest are medical parasitology, vector-borne diseases, and the pathology of infectious diseases. She has published more than 220 manuscripts on these topics, as well as 4 books and 36 book chapters. She shares her passion for medical parasitology in her Parasite case blog, Creepy, Dreadful, Wonderful Parasites, and on social media. She also hosts a weekly laboratory leadership podcast called Answers from the Lab. Dr. Pritt, welcome, and thank you for being here today.
Well, thank you, Dr. Welrich, for that really kind introduction. So I have no relevant disclosures for any of the information that I'll be discussing today, but I'll give my normal disclosure, which is that I'll be showing some disturbing information. The learning objectives for today are to discuss tips for successful implementation of New Year's resolutions. So Happy New Year. How many of you made New Year's resolutions? Oh, there's not a lot. Raise your hands. Come on, everyone in here. Okay, so that's very interesting. Okay, we're getting some hands raised online. Awesome, thank you. So we're going to talk about the ways to best keep those New Year's resolutions. And then we're also going to describe some biological mechanisms of parasites in relation to common human behaviors like keeping New Year's resolutions. And then we'll also talk about key lessons that you can learn from the science of parasitology with personal and scientific growth in mind. You can learn a lot from parasites that will help in your own personal and professional goals. And I'm going to take you on a journey, a case-based journey that will tie into some of these questions. But let's talk just a little bit about New Year's resolutions. You know, we've been doing these for a long time. Have you ever wondered where that came from? So the earliest written records goes back about 4,000 years ago with the ancient Babylonians. They celebrated their new year mid-March when they were planting their crops. So at the time, they would make promises to the gods, usually for repaying debts and good behavior, with the hope that the gods would bless them with good crop yields. So this continued then in ancient Rome about 46 BCE. Julius Caesar established January 1st now as the beginning of the year, because prior to that, it wasn't really a standard, and people would call the beginning of the year whenever they felt. So at this point, he named the first month of the year as after the god Janus, which is the god of beginnings. So Janus is an interesting god. He has 2 faces. He's usually depicted looking backward at the previous year and looking forward at the year to come. So here's a picture from what Janus would have looked like. Now, at this time, going into this new year, Romans would make promises to Janus, the god of beginnings, for good conduct for the years to come, in return, again, for blessings. Now, mostly these days, I would say that most New Year's resolutions are based on personal improvement and they're secular. So how many people make New Year's resolutions? According to the various polls, it's anywhere from 30 to 60% of US citizens. And there was this poll by Statica that actually showed that it was about 43% are not making a New Year's resolution. So that means that the majority of people are. What are their New Year's resolutions? Well, save more money. How many people had that on their New Year's resolution? Okay, eating healthier. Okay, exercising more. Losing weight. I was actually pleased to see that eating healthier is higher up than losing weight, because really if you eat healthy and you're eating right, then hopefully you'd be losing weight as part of that, in comparison to some of those fad diets that might lose weight but not really give you good health. And then there's more, taking up a new hobby, cutting down on drinking, decreasing social media use. So you could all put your phones away now. Volunteering more, traveling, being more positive, reducing stress, drinking more water, getting more sleep, doing more for the environment, and keeping your New Year's resolutions. Okay, so my first objective was to discuss tips for implementing your New Year's resolution successfully, because only about 10% of people succeed in keeping their New Year's resolutions. And that's kind of sad if they have really good resolutions. So some good goals for life in general, for anything, for keeping a goal: make a realistic plan, use SMART goals. And do you all know what SMART goals are? Specific, measurable, achievable, relevant, and time bound. Start small and keep it simple. Focus on one goal at a time. And I thought this was really interesting. Add instead of taking something away. So let's say you want to eat healthier, and maybe you're thinking that you shouldn't eat so much chocolate. I don't know, that's a tough one. But maybe you also want to start eating more vegetables. Well, it's a lot easier to say that you're going to eat more vegetables and start just introducing that into your diet. And by making it positive rather than punitive, taking away something you enjoy, you have a higher opportunity for success. Also, create a routine to facilitate habit formation. There are a lot of good books on how to create good habits. Now, once you make something a habit, like every morning you're going to get your cup of coffee and you're going to grab that carrot from the refrigerator and you're going to put it into your lunchbox, it just becomes part of what you do. Where's my carrot? I need to get my carrot while I'm drinking my coffee and carrots. I don't know. So also build a support system. Get a system where you get your spouse or someone that you live with to help you. If you're having your coffee and you didn't get out your carrot, well, okay, then your significant other is going to say, where's your carrot? And then celebrate your small wins. You know, if you did it every day for that first week, well, give yourself some sort of small celebration that doesn't undo all the good work you've done with the carrots. All right, so now that we've talked about our first goal, let's go on to our second goal. And this is really lessons that we've learned from Parasites, which is a case-based journey. I'm going to start with a case that we saw within the past year, 2 years or so. A middle-aged woman from Ethiopia who came in with several weeks of epigastric pain and intermittent abdominal cramping. And, okay, warning, I'm going to start showing some gross pictures. She passed a long worm-like object in her stool. And also, when asked, she said that she had passed several other worms in her stool on other occasions. So we got one of these worms to examine in the laboratory, and we got some additional information about her. She works as a pork processor at a local plant, not to be named. And this is the specimen that we received. And look closely. It is alive. Yeah. Okay, so when we manipulated this object, we were able to express these eggs. So this is a worm. And note that these eggs are round. They have these nice radial striations, internal hooklets. What is this? Taenia, yeah. So I was thinking people would say tapeworm, which obviously this is a tapeworm. Specifically, it's a Taenia tapeworm. All right, so Taenia species, this causes a disease called teniasis. This is intestinal infection with the adult form of the tapeworm. And there's 3 Taenia species that live in our gut, potentially. Taenia saginata, Psyllium, and Asiatica. Now, most people don't know they have this. But the good news is you probably don't get it in the US. Let me just say that so that people don't start getting nervous. We usually see this in immigrants and travelers. But for people who do have it, they are often asymptomatic and they usually don't know they have infection unless they pass the proglottids. And I note that it's active or passive passage. These things do move on their own. Sometimes they wriggle out on their own. Or sometimes they're just passed with the stool. Now, if you do have symptoms, it may be some abdominal pain, bloating, malabsorption, maybe some weight loss. Occasionally, those proglottids can end up in some place that they really shouldn't and cause appendicitis, cholangitis, or even pancreatitis from migrating proglottids. So let's take another picture of, or see another video of some moving worms. This is another Taenia species. This one's a bit more active. You can see that these proglottids, they're connected by a common nerve trunk, so they kind of move as one. But sometimes they break off individually, and then you get little individual ones that move like this. And they kind of look like little roundworms when they really come out. It looks like a little worm in and of itself. So people will find these on their undergarments or maybe in their bedclothes, and submit them to the laboratory. And we have many folks from the parasitology laboratory that can tell you stories if you'd like afterwards. Taenia, teniasis, we talked about when you have the adult tapeworm, but there's a complication called cysticercosis, a different form of the disease that can be seen with Taenia solium only. It's an infection with the larval form. And what happens is that you have someone with the intestinal tapeworm who is excreting eggs in their stool. And if they don't use good hand hygiene, those eggs can end up in the food and water supply, and people can unknowingly ingest them. And now they've ingested the eggs, which go throughout the body and cause cysts. That's cysticercosis. And unfortunately, the eggs of Taenia solium and Taenia saginata look the same. We cannot tell them apart, even though it's only Taenia solium that causes disease. So we have to look at the proglottids to differentiate them. We do that in the parasitology lab. We were able to find out that this patient has Taenia saginata. There was a little concern that she had Taenia solium working in a pork processing plant, but that was not the case. So she probably got this in Ethiopia before she came. So she was able to be treated with praziquantel, which is a fairly straightforward treatment, and she was presumably cured. And as long as she's careful about what she eats in the future, she won't become secondarily infected. All right, now were some of you thinking that maybe you could have used the tapeworms for your New Year's resolution? Because there is an association with tapeworms as a weight loss tool. All right, I'm here to tell you that it's not a good idea if you haven't figured that out yourself, but also that it's not really well supported by the science. So in the Victorian era and then early 20th century, 1900s, there were advertisements that promoted sanitized tapeworms, little cysts coming from meat of the beef tapeworm. So they would take them out of beef and sanitize them, whatever that means, and then put them in a jar, and you could ingest them in little capsule form and get your tapeworm. And it was a means to achieve a slimmer figure. And then the idea is that once the desired weight was achieved, you'd give the antiparasitic, and then the person would be cured of their tapeworm. So you'll see advertisements like this. This is a picture I took from the display on the House on the Rock. Have any of you been there? In Wisconsin. So it turns out this was a hoax. Is limited evidence that this really was used widespread. And this particular advertisement was shown to be a hoax, which I guess is a lot of the things at the House on the Rock are hoaxes. Anyway, there's this interesting book you can read by Zarzo et al., or this one is actually a manuscript, The Tapeworm and the Maria Callas Diet. Maria Callas was also suspected of taking tapeworms to lose weight. She was a famous opera singer. So dieting, another reading recommendation if you're interested is Calories and Corsets, A History of Dieting Over 2,000 Years. Why do we diet? Most of the time in our evolutionary history, we were trying to get enough food, but now we're in a situation with abundance. So the first evidence of dieting, and that use of the word diet, comes from the early Greeks and Romans. It actually comes from dieta, which means way of life. So your physician would prescribe a certain dieta, which would include health, wellness, exercise, food, and drink. But it wasn't really diet as we would know it. That wouldn't come until the Middle Ages with Old French and Middle English, when it turned into diet day, and it turned into more just a reference to food and drink. But nowadays, we use diet all the time. We still refer to it now as food, mostly, not usually drink. And it's often in the context of losing weight. Well, if you're interested in doing that, I would recommend the Mayo Clinic diet, which is actually quite good. Or you could buy the Mayo Clinic diet book. I haven't read it, but from what I've seen, the Mayo Clinic diet really has a focus on whole food, plant-based diets. So you're eating a lot of fruits and vegetables, things you can pronounce, not that hyper palatable, highly processed foods, carbohydrates, less protein and dairy. Beans are a good source of that as well. Fewer fats, fewer sweets. All right, now what if you wanted to take the Mayo Clinic diet with your carbohydrates and have a tapeworm theme without the tapeworm? Well, I have a solution for you. I found this fabulous pasta that is made from durum wheat with no true parasites in it. And I looked at this and I thought, oh my gosh, that looks like a parasite. Do you guys see it, or is it just me and I'm just seeing things? So I bought this, and this poor company would be horrified to know that I talk about this. But anyway, it cooks up quite nicely, looks just like the specimen containers we have in the laboratory. And, all right, so speaking of eating healthy, we're talking about New Year's resolutions, losing weight, not through tapeworms. Now, also, how do we eat healthier? Well, I have another case for you. A patient had cooked salmon and eaten it with her family, and it was then refrigerated and reheated for dinner the next day. The diners were dismayed to find a moving object in their reheated salmon. Okay, this case was donated by Dr. Sue Whittier. And yeah, look at what we see here. That's really unfortunate. So, all right, there we go. Nice unobstructed view. So here's a nice picture. So look at this salmon. You know, does this look a little undercooked to you? It looks a little undercooked to me. This part looks like maybe it was cooked on the outside, but there's this fresh, more undercooked portion in the middle, and that's clearly a worm. So this was not cooked well enough to kill the worms. So what is this worm? What is this condition? And this is not a, so I see a C from Peter. Excellent. Anyone else? This is C. That is the correct answer. This is anisakiasis. It's an infection you get by eating undercooked fish. These others are just distractors. They really wouldn't present like this. And actually, I should say that this patient thankfully did not ingest this worm. And so this could have led to anisakiasis, but didn't. We were able to catch it in time. So this is due to a number of roundworms that live in fish. And if you're interested in not getting this, then I would recommend inactivating fish by cooking it until it flakes easily with a fork. Or if you're going to eat it undercooked, like seared or raw, then you want to freeze it in a consumer grade freezer, like your freezer at home, for 7 days at -20. Or if you happen to have a deep freezer at home, you could freeze it at -70 for 24 hours and you'll be okay. So I will say that brining, smoking, marinating in lime juice, this does not reliably kill all parasites. If you want to make delicious ceviche, you really should either have a professional chef prepare it or freeze the fish well and then make it, or if you want to make pickled herring, for example. And thankfully, I know some people who are now following this advice. So you can do this safely. Unfortunately, some people develop allergies to anisakids, and then freezing and cooking doesn't help. They just can't eat fish anymore. And it actually makes me wonder how many people who think they have a fish allergy actually have an anisakiasis allergy. So if you want to read more about this, I like suggesting good reading material. This is from the FDA, and it talks about food safety, and it gets into all of the details about how you should cook food and at which temperatures. So let's do a fun little matching game and talk about why it's important to have food safety and eating healthy. And I have to say, I love these pictures. I created them on ChatGPT, actually on Dolly. That's a little bit more realistic than I think it probably would look, but those are frog legs. Okay, so let's start at the top. And just for the group that's in here, why don't you go ahead and shout it out? What parasite can you get by eating beef, undercooked beef? And the parasitology lab is allowed to shout it out. But residents, I'm looking at you as well. Tinea saginata, yes, nice. All right, what about pork? So Tinea solium, we just talked about that, right? But there's one other, yeah, trichinella. Pork is the #1 cause of trichinosis or trichinosis worldwide, although in the US, it's actually more wild game like bear and boar. Fish. Fish is a big one. Fish, as a natural creature, is usually caught in the wild. Farm-raised fish may have less parasites, but not necessarily. So wild-caught fish probably have multiple parasites. What are some of those that could be transmitted to humans? So we have anisakis. Yes, just talked about that. What else? Dibothriacephalus latus, previously known as Diphyllobothrium laudum. They had to change the name, I know. There's actually 2 more on here that could be transmitted by eating undercooked fish. Clonorchis. Nice job, Tom. Tom's an expert at this. There is 1 more, but it's a super tricky, super rare one. Maybe our micro fellows know. Spirometra. Causes a disease called sparganosis. All right. What about aquatic plants? Now, this has always been disappointing to me as a vegetarian that I could still get parasites and not just by eating aquatic plants like watercress and water chestnuts.
Yeah, Fasciolopsis.
Fasciolopsis busky and Fasciola hepatica, yes. All right, what about frog? You'll notice that these are all taken, but there is something on here that you could get by eating a couple different things, and frog is one of them.
I think maybe Spirometra can also be from frogs.
Yes, thank you, Adel. Nice job. So, be it frog, fish, pork, beef, or water chestnut, cook your food well or prepare it properly, and you'll avoid all of these parasites. So I think I fulfilled objective #2 of how parasites influence host behaviors like weight loss. And, we've talked about eating healthier, cooking your food, reading that FDA manual, losing weight, not with a tapeworm, but maybe with the Mayo Clinic diet. So let's move on to objective #3. Identify key lessons from the science of parasitology for personal and scientific growth. All right, this might be a little bit of a stretch, but we can learn a lot from parasites. And when we talk about things like adaptability, persistence, survival, we are undergoing a transformation here at Mayo Clinic. I think a very exciting, positive transformation, but it's a change nonetheless. So adaptability, persistence, survival are all important skills to have. Well, to emphasize this, I'm going to go back to the very first case I presented as a clinical microbiology fellow here back in July of 2006. So this was a 69-year-old female who presented to the emergency department who had this left-sided weakness. She had tripped over a cord walking across the floor, scraped her nose, maybe hit her left face. That wasn't clear. And at that point, when her family examined her and she was complaining about left facial pain, she was noted to have difficulty speaking and a left facial droop. So there was some thought initially of could that have been caused by fall, but no, that was a distractor. Now, her previous medical history was important in that she had a presumed polymyositis. That's an inflammation of the muscles. So she was receiving prednisone, so corticosteroid, and infliximab. Both of these can result in a person being immunosuppressed. So she's an immunosuppressed patient. This is a concern that puts her at risk for a number of infections. So they did a CT scan and then followed it up with an MRI because of their finding here of this 3.8 by 4.3 by 3.1 centimeter hemorrhagic mass in the right frontal lobe. Little bit of vasogenic edema, minimal mass effect. They were pretty clear that this was going to be either a primary malignancy, a brain tumor, or a metastatic malignancy. And the note specifically said that infection would be unlikely. It ended up being infection. I'm presenting it here today. But let's see what we have here. So we had brain tissue with inflammation. This is the leptomeninges. It was mostly lymphocytes. And then we had a lot of necrosis. And with the necrosis, they did not see any cancer cells, although that was a consideration. But instead, they saw these little structures here. And this is just with 4DX drive, what we typically see in histopathology. So what is your consideration? What parasite might be present here? Toxo. This is toxoplasmosis, still proven otherwise. So the sign out, actually, an immunohistochemical stain for toxoplasma did show positive staining. This was toxoplasmosis. So this patient was treated. She was started on drugs, pyrimethamine, sulfadiazine with vitamin leucovorin. Back in 2006, when I presented this, she was still in-house, but now I was able to go back into the medical record and find out what happened to her. And it turned out that she was discharged to a skilled care facility because she now has these new symptoms that are harder to manage with her family. But she lived for another 2 years and died of unrelated causes. So toxoplasmosis, it is caused by a very, very interesting protozoan parasite, Toxoplasma gondii. So it's a protozoan, it's a single-celled eukaryotic parasite, and the life cycle involves a cat of some sort, a feline. It could be a small cat like a domestic cat or a large cat, but it's a cat and mouse life cycle predominantly. So the cat is the definitive host where sexual reproduction occurs, and the cat sheds oocysts in the stool, which can then be eaten by a mouse. The mouse gets infected and forms these tissue cysts, and then the cat eats the mouse, and the life cycle continues. So the cat's key in this life cycle. But these oocysts get into the environment. They can be eaten by other animals like pigs, sheep, cows. And then if we eat those animals and we ingest those tissue cysts, then we can get infected. They could also get into the environment on contaminated fruits and vegetables and water. They can be in the litter box of the cat. So that's why they say pregnant women shouldn't scoop the litter box. And if you're pregnant and you get infected with Toxoplasma gondii, you could transmit it to your developing fetus. So that's really important to know. You could also get it by blood transfusion. So this is a really creative parasite in all of the different ways that it can infect the host. And it's a wide array of mammals and birds, essentially every single mammal and bird. So the turkey you had for Thanksgiving, maybe your Christmas goose, if any of you had that, that could have had toxoplasma. Again, going back to the FDA's guidelines, make sure you cook your food correctly. You're trying to avoid a number of things, including toxoplasmosis. Now, if you get infected and you're immunocompetent, you may not have any symptoms at all, or you may have a mild mononucleosis-like illness. What do I mean by that? Fever, swollen lymph nodes, feeling kind of off for a while, and then it goes away on its own. But for immunocompromised patients, it can cause life-threatening illness, and for the fetus as well. This is very important. So our patient that we just talked about, she had life-threatening illness. That would have killed her if it hadn't been treated, and then she wouldn't have had those 2 additional years with her family. So Toxoplasma gondii excels at persistence, adaptability, and survival. It infects virtually all warm-blooded animals and birds. It can be transmittable by all those different means, so transplacental, transfusion, ingestion of oocysts, ingestion of tissue cysts, number of different ways. It forms durable cysts, which probably survive for life in the host. So once you're infected, you're always infected. So if you happen to be infected as a child and later as an adult, you become immunocompromised for whatever reason, now you could have very severe disease. And it evades host immunity by downregulating inflammatory pathways and also creating a little safe haven for itself in these parasitophorous vacuoles. And I was able to work with our electron microscopy group, and it shows these nice little cysts with the bradiozoite parasite forms, and you can see them over here too. This is what they look like. This was a case that we had in our records, and I thought it would just be really informative to do electron microscopy on it. So the last aspect of toxoplasma I'm going to talk about that I think is particularly interesting is that this parasite manipulates the behavior of its host to ensure its own survival. So we talk about zombie parasites, parasites that take over its host. This is one of them. This parasite manipulates the host to make sure it has completion of its life cycle. Now, how do we know that? Well, there were some studies back in the 1990s, but this is a really nice one published in the year 2000 called Fatal Attraction in Rats Infected with Toxoplasma gondii. And with that, they actually had this experimental pen where they took rats and they infected 7 rats. And they also had 7 uninfected rats, and they observed them. They put them in this pen every night in different pens. This was a maze. And they had 670 hours of rat observation. They didn't have the person there. They recorded them from a distance. They cleaned the pens in between. And in the pens, they had 4 corners where they had different scents. They had bedding from the rat itself. So this should be considered safe bedding. Bedding from a rabbit, a non-threat, non-predator, neutral bedding that had no scent at all, and then they had bedding that had undiluted cat urine. Normally would have been a big red flag for a rat. And indeed, that's what they found. Uninfected rats had a strong aversion to cat odors. They recorded where this cat went, and the cat did not show any preference for this area and stayed away from it and tended to go towards the other areas. But the infected rats didn't. They had no avoidance. It's not that they went here more often. They just didn't care. They lost their fear of the cat odors, which is fascinating, especially because subsequent follow-up studies showed that this didn't apply to any other predators because they tried this with dogs and other things that could eat rats. They didn't lose their fear of them. It was just cats. And cats are the only definitive host for this parasite. Very interesting. Other studies have shown that these infected rats are more likely to have risk-taking behavior, reduced anxiety, increased sociability, all things that might make it more likely to get eaten. And they have some science behind this. It's probably due to structural changes in the amygdala, alterations in dopamine production, changes in gene expression. The question is, does this apply to humans? And this is very interesting. There are some studies, they're not definitive at this point, but there has been an association with certain diseases like schizophrenia. Again, not proven. So I think the jury's still out on that one, but also maybe more risk-taking behaviors like people that get into car accidents. Okay, so what can we learn from parasites like Toxoplasma gondii? Adaptability and flexibility. Persistence in its host for its lifespan, resource optimization, collaboration and symbiosis. We can learn that from other parasites. Sometimes parasites don't actually do a lot of harm. Overcoming barriers, efficiency, and goal-oriented behavior. This sounds like a checklist of things that you want to have for yourself. So I'm going to end there with my picture of my firework fleas jumping all over the place, and I would be happy to take any questions.
So Dr. Pritt, since you're so into history, what would it have been like during Julius Caesar's time as far as dealing with these things?
Oh, interesting question. You mean like just parasites in general? Well, first of all, I will say that parasites are one of probably the first microorganism to be described because many of them are big and visible. And if you go back to the Egyptian papyrus and even the Bible, there's references to things that are probably worms. So there's no question in Julius Caesar's time, they would have had some worms. They probably would have had intestinal parasites. They definitely had lice. In fact, louse combs for removing the nits had been found from about that same time period. Having said that, the Greeks and Romans had, at the peak of their civilization, pretty good standards of hygiene. So they probably would have kept these things at bay for the most part. But if you went out into the rural areas where they didn't have the aqueducts and the baths and the indoor plumbing, they probably would have had a lot more intestinal parasites, for example. The Romans also had and the Greeks had a very sophisticated cuisine with cooking. They loved their fish sauce, which is called garum. And they had, there's actually cookbooks from this time period that talks a lot about precise cooking conditions. So I don't know if it rivaled the FDA's cooking guidelines, but it was pretty good. So they probably weren't getting a lot of parasites from food when it was prepared correctly. But again, that's probably in the city, in the metropolis. And if you go out into the rural, poorer areas, I'm guessing that people had all those parasites then as well.
Any other questions from the audience?
Oh, Dr. Yao.
Go ahead, Dr. Yao online.
Hi, thank you, Dr. Pritt, for the lovely presentation. Luckily, I did have lunch before your presentation. I do have 2 questions. They are not related. First is relating to the salmon. Any evidence that farm salmon is less likely to be infected than the wild-caught salmon?
Yeah, that's a really good question, Dr. Yao. So farmed salmon, it depends on how it's farmed. If it's farmed in, like, say, a swimming pool type environment, like a controlled tank, then it probably is not ingesting the intermediate hosts and getting infected. Because you need the whole life cycle for Anisakis, it's actually marine mammals, so you probably don't have seals, etc., in that same environment. But if you have fish that are actually just in a netted off area in a natural body of water, then that's a different story. There's still that potential for contact with other wildlife. Having said that, the farmed salmon, because they're kept in very close conditions, they're actually more susceptible to things like fish lice, which I didn't know was a thing until I read about it more. And it eats away at their scales, and it's very devastating. So farmed fishing has its downsides, to be sure. But in terms of the wild, it is very, very common. About a third of all cod is thought to have anisakids. Any fish that is found in the wild pretty much has at least one form of parasite, because they're wild creatures and it's just a fact of life. In fact, as I said, for most of our evolutionary history, we had parasites as well, and still do have things like head lice and pinworms, even in our resource-rich environments here in the U.S.
But the actual parasites should be really rare if this is a big trawler, because they get frozen immediately. And
yes.
So that's
is that Dr. Grebe? Yeah, so they should be, if they're frozen, if they are solid frozen. If they're just thrown on ice, that might be a different story. Now, I will say, I didn't get into this, but the FDA, with their regulations for sushi and raw fish that's served, like salmon is served as sashimi and sushi, all restaurants that serve sushi and sashimi are supposed to apply the FDA guidelines, which would include that flash freezing to kill all nematodes, cestodes, and trematodes. And then they're also required to have that little warning in the menu that talks about the risk of eating undercooked food and dairy and seafood. But whether or not people follow that is unknown. And Dr. Yao, you said you had a second question?
Yes, I was curious about the medicinal leech therapy. I didn't realize that our pharmacy actually have a stock of these. We have seen reports of Aeromonas septicemia associated with use of these in the medical setting. I'm curious, how do we prevent that in the leeches that are provided here in our institute?
Yeah. Well, really good question. So the leeches, Hirudo medicinalis, which is actually thought to be probably a different species that we now have, but that's the medicinal leech. You get them from a set provider, distributor. St. Mary's orders those. We actually buy them at least once or twice a year. So we've been doing this for, I don't know, the past 15 years. So yeah, they definitely have this ready stock. The Aeromonas, unfortunately, is an obligate commensal in the leeches. You cannot get rid of it without hurting the leech. So you have to give prophylactic antibiotics if you're doing leech therapy to prevent the patient from becoming septic with Aeromonas. And I know of at least 2 cases where people got Aeromonas sepsis. Thankfully, it didn't occur at Mayo, but they were referred here afterwards. And actually, David Lynch, our previous supervisor of the initial processing laboratory, got the actual leeches that one particular patient had received, and then had the patient's isolate of Aeromonas, and he dissected the leech, which is not an easy thing because they're squishy and squirmy. And he got the Aeromonas of the patient and the leech, and he compared them. And back then we did protein electrophoresis and the I forget the name of the procedure, and we showed that they were identical. Pretty obvious, but yes, pulse field electrophoresis. Thank you. So that was interesting. So to answer your question, you can't get rid of it in the leech. You have to treat the patient preventatively.
Thank you.
It's like Carrie Severson.
Yeah, Carrie.
Yeah, I just was kind of curious, like your opinion about eating sushi in general.
Yeah, it's funny you say that. I'm actually now trying to do more of the whole foods plant-based diet with less of the fish, although historically, I've been a pescatarian. I will eat or had eaten some forms of raw fish. Ahi tuna in general is relatively safe. I stay away from salmon. To be honest, I will not eat salmon sushi. If it's prepared correctly, I'm sure it's safe and delicious. So I don't want to badmouth salmon at all. But I try to stay away from that now. I stick more with the cooked foods like eel and even just the vegetable-based sushis like the tamago, the egg is really good. Yeah, so that's my approach. But there are ways to safely eat a whole variety of foods. You just have to be familiar with what they are. Oh, Samantha.
Good afternoon, Dr. Pritt. Thank you. I was curious, you had mentioned how disappointed you were that as a vegetarian, you couldn't avoid parasites on marine plants. Are those parasites actually endemic to the plant specifically, or is that cross-contamination from like marine molluscids like snails, or do we know at all?
Oh, yeah, that's a really good question, Sam. So unfortunately, the plant is the host. They are one of the intermediate hosts. And so for Fasciolopsis busky and Fasciola hepatica, they have two intermediate hosts. The first is the snail. The second is the plant, which I guess, yeah, it's a living thing. It can be an intermediate host, just like a pig or a cow could be. So the parasite specifically insists in its metacercariae form on the watercress, on the water chestnuts, and then humans eat them. And if you don't cook them properly in endemic areas, you can get infected. So this parasite is present in some areas, Fasciolopsis busky of the Americas, more tropical areas. Fasciola hepatica isn't as widespread, but even still, you have to know in endemic areas, you don't want to eat the raw water chestnuts and watercress, which is too bad because it's delicious.
And also, we should be very proud of our parasites because the medical profession has it as its emblem, haven't we?
Yeah, so Dr. Grebe, that's an interesting point you bring up. This is actually one of the things that got me interested in parasitology to begin with. So the staff of Asclepius, we say that wrong. It's the staff with the serpent or the worm wrapped around it. There's some thought that actually came from Dracunculus medinensis, the guinea worm, that to this day we have to remove by winding it slowly around a stick. Unfortunately, that now has been debunked, that this is present before we really even knew about Dracunculus. But back in 1994, I took a course on some sort of evolutionary biology, and I learned about this. And I never knew I was going to become a parasitologist, but that really inspired me at that time. And of course, you're all familiar with the caduceus as well. That's the medical staff with two serpents. It's actually not really associated with medicine, but it had been inadvertently associated. So it's probably a snake, but still very interesting.
The military didn't know that that it's a different one.
I know, the military messed up. So they used the two-headed serpent, which was not meant to be. It was supposed to be the staff with the single serpent. Anyway, yeah, I love that history, Stefan.
Yes. And
All right.
there's so many. I mean, there's so many interesting things with parasites. I mean, if I wasn't a fan of endocrinology, I might have become.
You'd make an excellent parasitologist, Dr. Grebe. I could talk about this all day.
Well, thank you so much, Dr. Pritt, for a fascinating and engaging talk.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.